All right. All right. Hello, everybody. We'll just maybe give it 1 or 2 more minutes because there are also quite a few of folks still coming in. Um, and just get this started. All right, cool. Let's get the show started. Welcome everybody to the 2025 Biology of Reproduction Paper of the Year webinar. We have many registrations and now we are having 100 plus participants and the number is still increasing. What a way to kickstart the seminar series for this academic year. So this webinar is organized by the SSR Virtual Education Committee. My name is Hai Chi Chen. I'm a faculty member at UT Southwestern Medical Center in Dallas, Texas, and I will be your moderator today. So before I introduce our speaker, I just wanted to go quickly through a couple of housekeeping items as people are still coming in. So the first thing is that this webinar is about 40 minutes to 45 minutes long, followed by 10 to 15 minutes for Q&A. And then we encourage you to use the Q&A feature underneath in your Zoom window so that you can tap in your questions as the webinar continues. And then this webinar will be recorded and it's actually being recorded right now. And the link to the recorded content will be shared with everyone who has registered in a couple of days. So look out for that email. And so now let's go to the main show. And it's my great pleasure to introduce Dr. Greg Johnson. So Dr. Johnson is a professor in the Department of Veterinary Integrative Biosciences at Texas A&M University. He's also the Assistant Dean for Research and Graduate Studies in the College of Veterinary Medicine and Biomedical Sciences. So Dr. Johnson received a bachelor's degree in zoology and a master's degree in microbiology and a PhD in reproductive biology from the University of Wyoming. That's a quite a mixed background you have here. So following PhD graduation, he went on to do a postdoc with Dr. Fuller Baser within the Department of Animal Sciences at A&M, and then became a faculty member of the Department of Animal and Veterinary Science at the University of Idaho before moving back to A&M to be a full professor. His primary research interest now is pregnancy, with an emphasis on the interactions between the uterus and the conceptus that mediate the establishment and maintenance of pregnancy. This includes pregnancy recognition, conceptus implantation, placental development in farm animals such as pigs, sheep, and cows. Dr. Johnson has received numerous awards both on the research side as well as on the teaching side at his university. And he's also served on the board of directors for SSR. And I just learned that he was also a member of the Heritage Committee. So thank you so much for your service, Greg. And now back to you. Looking forward to your talk.
Can you see it? Is it in presentation mode?
Yeah, looks perfect.
All right, well, thank you for that introduction. And I'd like to start out with a few other thanks before I get into the meat of this presentation. I would like to thank Biology Reproduction for the recognition. I'd like to thank the USDA, who has funded all the work that I'll show today. I'd like to thank several of my very close collaborating scientists, including Fuller Baser, Robert Burkhardt, Iwan So, Gaia Wu, Gunter Wagner, Claire Stenhouse, and Kai Poehler. And I particularly want to thank several of the trainees whose work will be shown in this presentation, and a special shout out goes to Dr. Joe Kane, who was the first author of the paper that was given the recognition. Now, something all of us have in common is an interest in pregnancy. And complications in pregnancy are common across all the mammalian species. I kind of believe that if you lumped all pregnancy losses into one group as a disease, it would be the most formidable disease that we face. Certainly, there's the heartache of couples trying to have babies. I don't think anybody tries to make mice reproduce better, but they're a very valuable tool for looking at pregnancy in a mechanistic manner. But the species I'll be focusing on today, and I decided to spread it around to the 3 species rather than focus completely on the pig, and that'll be the cow, sheep, and pigs. These are the livestock species. And we're talking about the livestock species, pregnancy loss is economic loss. So I'd like to step back and just remind you of the histology or microanatomy that I'll show in multiple slides today. In most cases, these will be paraffin embedded cross sections of the uterus or the placenta or conceptus and the uterus. The conceptus and placenta will be to the top usually, the uterus to the bottom. And when I use the term conceptus, let me get this pointer correct here. Use this term conceptus, that means the embryo, fetus, and associated placental membranes, but I'm only going to be showing associated placental membranes. In all cases, they'll be immunofluorescent stained, usually for some particular protein, and stained with DAPI to look at the nuclei. And I've chosen 3 days of pregnancy from the pig. Day 13, when the conceptus is not attached or opposed to the uterine luminal epithelium. Day 15, when the conceptus is closely opposed to the uterine luminal epithelium, but can still be flushed out intact. And day 25, when the conceptus is firmly attached to the uterine luminal epithelium. And this has been immunofluorescent staining for PCNA, so we're looking at proliferation. And I think these 3 panels illustrate that localization of a single protein can really provide a great deal of information about the physiology that's going on. Because we see here at days 13 and 15, the conceptus is highly proliferative. And it's highly proliferative because it is elongating. By day 25, it's become a little less proliferative, but it is still proliferating because that interface is going to fold quickly after this. We see that the luminal epithelium, in sharp contrast, is not proliferating on days 13 and 15. It needs to be a stable base for this conceptus to attach to for implantation. However, by day 25 of gestation, the luminal epithelium is highly proliferative, and that is because it's preparing to fold the uterine placental interface, which is a characteristic of its epithelial chorioplacentation. Interestingly, in the pig, these early days, the glandular epithelium is not proliferative. This is different than what you'd see in sheep and cattle. And I want you to note the proliferation that's occurring in the endothelium of the vasculature of the stratum compactum stroma, which is this vasculature can provide many of the nutrients that will be transferred through the luminal epithelium into the lumen to support these conceptuses' development before they attach to the uterine luminal epithelium. Now, to get to the actual data, I'd like to begin by talking about conceptus elongation, and I'm going to focus on some work that we've done in the pig, but along with Dr. Fuller-Beza, we've done much of this work in the sheep as well. And we know that conceptus elongation is required for pregnancy recognition, expression of conceptus interferons, and implantation. Now, the livestock species are similar to all species when they form a round blastocyst, and you can see that at day 10 of gestation here. But after that point, they differ from what you see in rodents and humans, where they'll hatch from the zona pellucida and then they will elongate. They'll form tubes. We can see those tubes here on day 10 of gestation. They will elongate. We'll see these elongated forms at day 11 of gestation here, but they don't stop there. They keep elongating until they form these long, fragile filaments. Now, this change in morphology, this change in development requires a great deal of cell proliferation, migration, and changes in morphology. And this requires the energy in the form of ATP. Well, we believe that the proliferation, migration, and morphological changes of Conceptus trophectoderm is supported by ATP that is derived from the hexose sugars, at least in part. We know that glucose and fructose increase in the uterine lumen during the peri-implantation period. Glucose and fructose move into and out of cells through their SLC transporters. And this is an in situ hybridization for SLC2A3, a glucose transporter. And we can see its mRNAs in the trophectoderm. This is immunohistochemistry for SLC2A8. This is a glucose and a fructose transporter. And we see it's expressed in the trophectoderm as well. And we know that these elongating porcine conceptuses can metabolize glucose and fructose. We took day 16 conceptuses that are elongating and incubated them with radiolabeled glucose, fructose, or the combination. Then we looked for the CO2, for radioactivity in the CO2 that was produced upon metabolism of the sugars. And what we found if you give them glucose and fructose alone, they'll metabolize both sugars at about the same rate. But if you give them glucose and fructose together, they preferentially metabolize glucose over fructose. So again, this is PCNA staining, immunostaining, and Western blotting. These conceptuses during the peri-implantation period are highly proliferative, they're highly migratory, and they can metabolize glucose and fructose. When you go into the literature, you see that highly proliferative migratory and immune and tumor cells will switch glucose metabolism from oxidative phosphorylation to glycolysis. What they're doing is they're performing aerobic glycolysis, or the Warburg effect. This is going to produce only moderate amounts of ATP, but it will produce NAD, which can be used to maintain glycolysis, and lactate can be funneled back in through glucose to maintain glycolysis. And it's important to maintain glycolysis in proliferating cells because it optimizes your ability to glean glycolytic intermediates off of these side chain biosynthetic pathways. And these intermediates can leave glycolysis to take part in diverse biosynthetic reactions needed for the cell proliferation, migration, and morphological changes. The pentose phosphate pathway, you're going to get ribose, hexosamine biosynthesis. These are going to be phosphorylation events. And you can get the production of amino acids such as serine, which can be funneled into one carbon metabolism for the productions of purines and thymidine for DNA synthesis. Now, glycolysis coupled with the TCA cycle is a great way to get ATP, but you do use up the NAD needed to maintain glycolysis, and you don't get the production of lactate. So our first question is, do these conceptuses perform aerobic glycolysis? So we incubated the conceptuses with glucose and fructose, and indeed they did produce lactate. So they have an active aerobic glycolytic pathway. Next, we wanted to look at some of these side chain biosynthetic pathways, and the first one we looked at was one carbon metabolism. In one carbon metabolism, 3-phosphoglycerate is pulled out of glycolysis, and through the consecutive actions of PHGDH, PSAT, and PSPH is converted to serine. Serine is then involved in the conversion of tetrahydrofolate to formate, which can eventually produce the purines and thymidine. Now, we know our porcine conceptuses develop in a somewhat hypoxic environment. Hypoxia can turn on mTOR in cells, and mTOR can increase HIF1 alpha, which will increase the activity of SHMT2 and MTHFT2, which are required to make formate. So we want to look at some of these factors at the uterine placental interface. And what we found is that PHGDH, PSAT1, and PSPH are made by the luminal epithelium. SHMT2 and MTHFT2 are made by the conceptus trophectoderm. We have active mTOR pathway in our conceptuses because we can see phosphorylate RPS6, and we have high expression of HIF1 alpha in the conceptuses. We then incubated day 15 and 16 conceptuses with radiolabeled serine, and we're able to make radiolabeled formate. So this is an active pathway, and what appears to be happening is it's the luminal epithelium of the uterus that converts 3-phosphoglycerate to serine, secretes it into the uterine lumen, where it is then confronting these conceptuses that have been primed by mTOR and HIF1 alpha to respond to the one carbon metabolic pathway. So it's an active pathway, but does this have anything to do with conceptus elongation? And to answer this question, we installed mini osmotic pumps and infused methotrexate into the uterine lumen of day 10 pregnant pigs. Now, methotrexate blocks the formation of dihydrofolate reductase, and dihydrofolate reductase is required to make tetrahydrofolate. So this then negates one carbon metabolic pathway. In these experiments, we took the uterine horn from day 10 pregnant pigs, and that's just prior to elongation. We ligated them about a third of the way from the uterotubal junction to the uterine body. This is to minimize the surface area that we're pumping into. We then took a catheter, threaded it through the uterotubal junction, connected a mini pump containing methotrexate, and methotrexate was then pumped into the uterine lumen for exposure to these elongating conceptuses. When we pumped in control proteins, we didn't affect conceptus elongation, but when we pumped in methotrexate, we delayed or slowed conceptus elongation. So it appears that these elongating conceptuses do utilize one carbon metabolism. Another interesting pathway is the pentose phosphate pathway, and the key enzyme in the pentose phosphate pathway is glucose 6-phosphate dehydrogenase, and it is indeed present in conceptus on the mRNA and protein level. And when you localize it, it's localized to the conceptus trophectoderm on days 15, 18, and 25 of gestation. When you co-localize with pCNA, most of the proliferating cells express G6-PDH. We asked, is this an active pathway in our elongating porcine conceptuses? And to do this, answer this, we incubated these conceptus day 15 and 16 with radiolabeled glucose and fructose. But we did it in two ways. We labeled the first carbon, which goes down through glycolysis, but it'll also go into the pentose phosphate pathway, or we incubated them with labeled on the sixth carbon, which goes through glycolysis, but can't get into the pentose phosphate pathway. We then measured the radioactivity produced in the CO2 upon metabolism of the sugars, subtracted C6 from C1 value to find out how much was going into the pentose phosphate pathway. And what we found is glucose is pretty good at sending carbons into the pentose phosphate pathway, but fructose not so much. So we have these elongating conceptuses that are converting pyruvate to lactate for aerobic glycolysis. They have an active pentose phosphate pathway. They have an active one-carbon metabolic pathway. And this then will likely result in a lack of pyruvate to feed the TCA cycle. And these conceptuses still need to have ATP in order to elongate. Well, we know cancer cells replenish the TCA cycle through what's called anaplerosis. And these are pathways through which TCA cycle intermediates other than acetyl coenzyme A can be supplied. The major anaplerotic substrate in proliferating cells is glutamine, and this is called glutaminolysis. Now, in glutaminolysis, glutamine can be converted to glutamate through the actions of glutaminase, a reversible reaction with glutamine synthetase, and glutamate can be converted to alpha-ketoglutarate, which is a component of the TCA cycle via the aminotransferases or GLUT1. We know from previous work from Dr. Fuller-Bazer's laboratory that they have an increase in glutamine in the uterine flushings of pregnant pigs during the peri-implantation period. And if you treat porcine trophectoderm cells with glutamine, you'll increase their proliferation. So we want to look at some of these factors at the uterine placental interface. And indeed, glutaminase mRNA is present in these conceptuses. The protein is localized to the conceptus trophectoderm. Interestingly, glutamine synthetase is in the endoderm. And each of the aminotransferases, PSAT1, GOT2, and GPT2, increase in elongating conceptuses, but GLUT1 decreases. So these conceptuses have the machinery to convert glutamine to glutamate, and then glutamate to alpha-ketoglutarate using the aminotransferases with the added addition of production of amino acids. But once again, is this active in these elongating conceptuses? And to answer that, we incubated day 15 elongating conceptus with radiolabeled glutamine. But we did it in two ways. We did it in the presence of glucose or in the absence of glucose. Now, if you've got glucose in the media, there's always going to be pyruvate present. You don't have any pressure to turn on glutaminolysis. But without the glucose, you're going to run out of the pyruvate. And now we see if these cells can really activate glutaminolysis. And indeed, they could. They increased their incorporations of carbons from, they increased their metabolism of glutamine in the absence of glucose. Now, this data is similar, except it's got a different endpoint. In this case, we're looking at the incorporations of carbons into intermediates of the TCA cycle, and those carbons are coming from glutamine. And what we found, we had an increase in the incorporation of carbons from glutamine in glutamate, alpha-ketoglutarate, succinate, malate, citrate, and aspartate. So these elongating conceptuses, they're going through glycolysis. They convert pyruvate to lactate for aerobic glycolysis. They have an active pentose phosphate pathway. They have an active one-carbon metabolic pathway. And to maintain TCA cycle flux, they are able to activate glutaminolysis. Now, this elongation is essential. It's essential for successful secretion of factors required for pregnancy recognition and implantation. So the peri-implantation period of our livestock species is pretty complicated. It's complicated for all species, but might be a little more for our species because they remain in the uterine lumen not attached for such a long period of time. You have the production of histotroph becoming from the glands and the luminal epithelium. And that histotroph acts to maintain and nourish and help the development of these elongating conceptuses. Eventually, these conceptuses elongate enough that they start producing paracrine factors that act back on the endometrium. Now, some of these paracrine factors act on the luminal epithelium and shallow glandular epithelium to block the luteal pulses, prostaglandin F2 alpha for pregnancy recognition. But there are a array of other conceptus-produced paracrine factors that can affect the function and gene expression in the endometrium. And that is very nicely illustrated in the Spearman correlation of endometrial gene expression in the pregnant pig provided by Dr. Gunther Wagner, where we're looking at days of pregnancy 13 to 25 from left to right and top to bottom. It's very clear that gene expression from day 13 to 15 is very different than gene expression from day 20 to 25. There's a major change in gene expression in the endometrium between days 15 and 20, and we think that is due to the production of several of these paracrine factors acting on the endometrium. Well, we know pigs, sheep, and cattle, conceptuses all produce interferons that upregulate interferon stimulated genes in the endometrium. And that's illustrated here. Here we're looking for immunofluorescence staining for interferon tau in the cow conceptus, the sheep conceptus, and interferon gamma in the pig conceptus. And down here we can see the upregulation of interferon stimulating genes in the stratum compactum stroma. In this case, for the cow, it's ISG15. For the sheep, it's STAT1. And for the pig, it's class 1 MHC. Now, to the left here is an in vitro cell culture experiment that illustrates that STAT1 and STAT2 are not only intracellular signaling molecules for the interferons, but they're also interferon-stimulated genes. What we did is we took endometrial cells from a sheep, incubated them with interferon tau, and within a half hour, we saw translocation of all of these STATs, which we expect they're part of the intracellular signaling for interferon tau. But by 24 hours, we saw upregulation of STAT1 and STAT2 in the cytoplasm of the cells, indicating that they are indeed interferon-stimulated genes. At the right is the general pattern of expression for interferon-stimulated genes in the sheep uterus, and this is for ISG15. It's in situ hybridization for mRNA. You find it in luminal epithelium on day 11, but by day 13, it's downregulated in the luminal epithelium due to IRF2. It upregulates in the stroma at that point, and it increases in stroma through day 15 of gestation. Now, this is interferon tau, produced by a sheep conceptus, but I want to focus on interferon gamma and the pig for this presentation. And certainly the pig conceptus produces its array of paracrine factors, and some really nice work from Rodney Geisler's laboratory in Missouri has shown when he knocks these out of the porcine conceptuses that estrogen, interleukin-1 beta, interferon gamma, and PGE2 are all essential to maintain conceptus development through the peri-implantation period. But we know that interferon gamma is the second most highly upregulated gene in the elongating conceptus, and you can see it here expressed in the trophectoderm on days 15 and 18 of gestation. It's starting to downregulate at day 20. And this interferon gamma acts in a paracrine manner on the endometrium. This is in situ hybridization, and we're looking here at the films of serial sections, so we can see the entire cross-section of the uterine wall. Here we have interferon gamma, and here we have STAT1. And interferon gamma is expressed by the conceptus, STAT1 by the endometrium, but only in regions adjacent to the conceptus, not in other areas of the endometrium, strongly suggesting that interferon gamma acts in a paracrine manner to increase STAT1, this interferon-stimulated gene, at sites of implantation. Now, they've been doing explant cultures for years with interferon tau in sheep and bovine cyclic endometrium, but this can be done as well. It's been done for years by Hak-Young Kao's laboratory. You can treat explant cultures from cyclic pigs with interferon gamma and get the upregulation of interferon-stimulated genes. And here's just 4 examples from the immune system. So what we're seeing here is an overview of an implantation site and a non-implantation site from day 15 of gestation in the pig that's been immunofluorescence stained for interferon gamma. And we think this picture implies local transport of interferon gamma from the conceptus into the endometrial luminal epithelium. And this is how the argument goes. At day 13 pregnancy, we see that the interferon gamma is in the conceptus, but not yet in the luminal epithelium. But by day 15, we have interferon gamma in the conceptus. We don't have interferon gamma in the luminal epithelium at inner implantation sites, but now we have interferon gamma in the luminal epithelium adjacent to the implanting conceptus. And it's not just at the apical surface like it's bound to a transmembrane receptor. It's found throughout the cell and often deposited right at the base of the cell, which is right next to the stroma where these interferon stimulated genes are going to upregulate. When we went back to our old in situ hybridization for interferon gamma, we never see interferon gamma mRNA in the luminal epithelium. The pig luminal epithelium doesn't make interferon gamma, so this interferon gamma must be coming from the conceptus. And we made the assumption, not the assumption, the hypothesis that it was being carried there by extracellular vesicles and that they could carry it through these cells. So Joe did what you would expect. He did uterine flushings from the cycle and pregnancy. He looked for some of the markers for EVs, and the EVs were there. They're both cycle and pregnancy. The endometrium makes lots of EVs, but interferon gamma was only present during the pregnancy. He then purified these EVs and found that they increased in size and number between days 11 and 15 of gestation. And then he took those EV preps and he did a Western blotting for interferon gamma, and day 15 of pregnancy is where he found the interferon gamma. He then took these preparations and treated explant cultures from cyclic pigs. And he found that these two very classic interferon stimulated genes, MX1 and OAS, this gamma in the EVs is actually better at upregulating these interferon stimulated genes than recombinant interferon gamma. But Joe looked at a whole array of other known interferon stimulated genes, and he found that in all cases, these EVs containing interferon gamma can upregulate interferon stimulated genes in explant cultures of cyclic pigs just as well as recombinant interferon gamma itself. And then he localized the interferon gamma in these explants. You do find some interferon gamma in the luminal epithelium of these explants, but it's more apical and it's not highly expressed, but you find a much more robust presence of interferon gamma in the luminal epithelium of explants incubated with EVs containing interferon gamma. So we believe EVs increase transport of interferon gamma into the endometrial luminal epithelium. So we have these conceptus interferons, and they're upregulating interferon stimulated genes in the stratum compactum stroma. But is there any physiological relevance to this? Well, one thing we do observe is we see an accumulation of immune cells in the endometrium during the peri-implantation period. We did immunofluorescence staining for CD45, which is a pan immune cell marker, and indeed they accumulate during days 15 and 18. Then we did immunofluorescence staining for tunnel and found that there's a modest increase in number of cells undergoing apoptosis at day 15. Now, these are both signs of inflammation. And when you're talking about inflammation, T cells come to mind. So we looked for T cells, looking at CD3, and we found indeed there is an increase of CD3 positive T cells on days 15, 18, and 20 of gestation. And if we co-localize this with pCNA, you see that many of the T cells are proliferating. Now, proliferating T cell is an activated T cell. It's an activated T cell. And to activate a T cell has to use its T cell receptor to recognize antigen in the context of class 1 MHC on an antigen-presenting cell. But it also must use a co-stimulatory molecule to recognize a receptor, to recognize a co-stimulatory ligand on the APC to completely become activated. So we looked for these ligands on APCs and receptors on T cells by RNA in the endometrium. And we found one of the ligands on APC, CD80, increased during the peri-implantation period, but all of the receptors on T cells increased during the peri-implantation period. And when we infused interferon gamma into pseudopregnant pigs using the mini pumps I talked about previously, we couldn't do much with the ligands on IPCs, but we did increase PDCD1, ICOS, and CTLA4 was strongly trending towards increasing. If we'd had a few more animals, I think it would have. We then looked for chemokines that could call these T cells into the endometrium, and we found that CXCL9, 10, and 11 all increased during the peri-implantation period. And when we infused interferon gamma into the pseudopregnant pigs, we could increase CXCL9, and CXCL11 was trending towards increasing. Finally, Brian McClendon performed fluorescence activating cell source to figure out what subpopulations of T cells are in the endometrium, and he found that most of these are CD4 positive T helper cells. Although we do have some CD8 positive cytotoxic T cells, some CD4 positive CD25 positive T regulatory cells, and some CD4 positive CD8 positive naive T cells. And this is one of those cases where it's really nice, because just a year later, Rodney Geisert published his work on knockdown of the interferon gamma from pig conceptuses, and one of the first things observed was a reduction in stromal CD3 positive cells. So we hypothesize, we believe that conceptus-derived EVs likely mediate the transport of interferon gamma into the endometrium to increase expression of interferon stimulate genes and recruit T cells that prepare the uterus for implantation. Now, one of the things Rod noticed when he knocked out interferon gamma is that these interferon gamma knockout conceptuses attach to the luminal epithelium, but they do not complete implantation. Now, in our livestock species, implantation is considered to be an adhesion or attachment cascade. You have pre-contact periods, apposition periods, and adhesion periods. You get downregulation of anti-adhesive molecules from the apical surface of the luminal epithelium, which then exposes the apical surface of the luminal epithelium to the apical surface of the trophectoderm for binding interactions. And most of these early binding interactions are via carbohydrates, lectins, and glycans. And Dr. John Applin has done a lot of great work with that in other species. But later, it is sealed down with integrins binding to their extracellular matrix proteins. Now, shown here at the left is an implantation site from a day 20 pig, day 20 sheep, day 26 cow, and day 31 cow. Now, I don't have time to go through these in detail today, but you can find these pictures if you look up this reference. I started in implantation, over 20 years ago with this Western blot. And what we saw in this Western blot was an upregulation or increase in osteopontin in the uterine flushings of pregnant over cyclic use. Now, it's been over almost 30 years since Bob Burkhardt established that integrins are prominent at the uterine placental interface of pigs, and we wanted to expand that to sheep and include a matricellular protein that binds many integrins called osteopontin. Now, osteopontin is a complex guy. He's also known as OPN, secreted phosphoprotein 1, or SPP1, and he has other names as well. We established that it does increase in expression during the peri-implantation period in the endometrium. And as I showed you before, you increase the protein in uterine flushings in pregnant over cyclic animals. Now, in general, osteopontin is just a single chain polypeptide, but don't let that fool you. It's pretty complex. It can be that goes through a lot of post-translational modifications that can change its function. It's a notoriously hard protein to work with in many areas of physiology. It can be variably phosphorylated, variably glycosylated. It can be cleaved into bioactive free fragments by the serine proteases as well as the MMPs. But what I'm going to focus on today is its ability to use this RGD sequence to bind to multiple integrin heterodimers. Now, these integrin heterodimers are transmembrane, non-covalently linked heterodimers composing of an alpha chain here in the orange and a beta chain here in the blue. Integrins integrate information from the outside of the cell to the inside of the cell and vice versa. Therefore, when a matrix protein like osteopontin binds an integrin, they have the opportunity to profoundly influence functions in that cell. One of those functions could be attachment for implantation. Well, osteopontin is highly expressed by the glandular epithelium of the sheep throughout gestation. It actually increases earlier than it's about 13 to 15 of pregnancy, and this is in situ hybridization for mRNA. It's a secretory protein, as SPP1 implies. If you overectomize use and treat them with progesterone, or treat them with progesterone and ZK, a progesterone receptor antagonist, the progesterone recapitulates what you see during pregnancy, and ZK completely ablates this. But note two things. We don't see any osteopontin mRNA on the luminal epithelium, and stuff I'm not showing you, we don't see it in the trophectoderm either. But we see osteopontin protein on the luminal epithelium and the trophectoderm. And we know we have an increase in its secretion into the uterine lumen. So that came up with the hypothesis that osteopontin is upregulated in the glandular epithelium by progesterone, secreted into the uterine lumen, where it binds back to the apical surface of the luminal epithelium and to conceptus trophectoderm, presumably via integrins. And this is still a very real possibility, because if you do immunofluorescence staining for MUC1, that anti-adhesive molecule, it's highly expressed at the apical surface of the luminal epithelium during early ester cycle, but downregulates in late ester cycle and during early pregnancies. And we get the expression of multiple alpha and beta integrin subunits at the uterine placental interface, many of these remarkably co-localized with osteopontin. So if osteopontin is binding integrins on these ovine conceptuses, we asked, well, what integrins are they binding to? And to answer this, we took ovine trophectoderm cell membranes and we performed chromatography for osteopontin binding, and we found that there were alpha and beta chains of integrins that bound to osteopontin from the plasma membranes. We then took these fractions and performed immunoprecipitation using multiple integrin antibodies, and it's the alpha V beta 3 integrin that osteopontin is binding to on these trophectoderm cells from sheep. We then performed a whole array of adhesion assays. We used BSA as a negative control, human fibronectin as a positive control, and two forms of intact osteopontin. One here where it's purified from bovine milk, and the other is osteopontin that's recombinant rat with an intact RGD binding, integrin binding sequence. And if you have intact osteopontin, it supports very robust trophectoderm cell adhesion, much better than human fibronectin. And it appears to be integrin mediated, because integrins require these divalent cations to bind a matrix protein. But if you perform these experiments in the absence of divalent cations, you greatly reduce osteopontin's ability to support trophectoderm cell adhesion. And the same is true if you perform these experiments in the presence of the alpha V beta 3 integrin binding antibody. So we believe osteopontin binds alpha V beta 3 integrins on trophectoderm cells to support cell adhesion. But we're looking at integrins and we're looking at osteopontin, and cell adhesion is a pretty simplistic endpoint for this biology. Because when a matrix protein like osteopontin binds to integrins in a membrane, it will cause those integrins to aggregate within the membrane and then associate with a whole array of cytoskeletal elements and signaling molecules within the cytoplasm. These aggregates on both sides of the membrane are so large that they can be detected by immunofluorescence staining and have been called integrin adhesion complexes. They used to be called focal adhesions. Over here on the left are trophectoderm cells that have bound down to osteopontin coated on a slide. And we see these integrin adhesion complexes have formed at the base of these cells. We know they're integrin adhesion complexes because we've got beta 3 integrin on one side of the membrane and talon on the other side of the membrane. Now, this is at the base of these cells as they attach to the osteopontin on the slide, but our hypothesis is that these integrin adhesion complexes can form at the apical surface of these cells and bind osteopontin for attachment to luminal epithelium for implantation. And we really needed to illustrate that this was a possibility at the apical surface of these cells. To do this, we took ovine trophectoderm cells and we incubated them on collagen. We then took polystyrene beads, we coated them with osteopontin, layered them onto the surface of the cell. The hope was that if there's integrins in that apical surface, they will bind to the osteopontin on the bead, aggregate, and we could detect the integrin adhesion complexes with that aggregation. Then we did immunofluorescence staining for red, talon for red, alpha V integrin for green, and if you had an alpha V integrin involved focal adhesion, it should co-localize as yellow. We took optical slices from the base of the cell up through to the apical surface. This is the base of the cell. We have all these focal adhesions at the base of the cell, just like what we saw previously on the previous slide. In this case, they incorporate talon. There's no green down here because alpha V integrin does not bind collagen. It's very interesting here that just the weight of this bead, that torsion, has caused more focal adhesions to form beneath where the bead is on the cell than at other places. But as you move up through the cell, you start to see the green form around the bead, and you get the co-localization of yellow, strongly suggesting that osteopontin induces in vitro integrin adhesion complexes at the apical surface of trophectoderm cells. Now, these are in vitro focal adhesions, but we see similar aggregates of integrins at the uterine placental interface of sheep throughout gestation, but we see them in interplacentomal regions, not so much in the placentomes. And I won't have time to talk about why I think that is, but if you want to ask later, that's fine. And these are big events in the interplacental regions of placentation. So you sometimes get aggregation a third of the way down through the cell. Sometimes it's on the luminal epithelium, sometimes on the trophectoderm, sometimes both. These are very reliable to find and can be spotted at the lowest objective. So I'd like to conclude this presentation today by just going through some observations that we've made concerning the uterine placental interface during early gestation in cattle. And these observations led to a currently funded USDA grant for Siwan So, myself, and Kai Poehler. Now, it's completely agreed and accepted that in the trophectoderm layer cattle, you have binucleate trophoblast giant cells that express PEG form. These will then fuse with luminal epithelial cells to form trinucleate fetal-maternal hybrid cells that are PEG positive. And certainly this occurs. We've published it ourselves. This is a binucleate cell with green showing PEG binding to a luminal epithelial cell with red for E-cadherin. And here's a trinucleate cell. You can't hard to see those nuclei in there that is expressing PEG. And here we have a cell that's trinucleate, and you can see the PEG is distributed to the basal surface, presumably to be secreted down to the vasculature of the endometrium. And that is what is seen in the mature placentome, as is drawn out and diagrammed very nicely here by Dr. Christiana Ferrar. Indeed, in these pictures here, we see a binucleate PEG positive cell in the trophectoderm and 3 trinucleate PEG positive cells in the luminal epithelium. But when we look closely, we find there's a more varied population of PEG positive cells at the uterine placental interface of cows than is predicted by this model. There are mononucleate PEG positive cells in the trophectoderm, and we're not the first to show this. This has been previously published, and mononucleate PEG positive cells in the luminal epithelium. Further, we can find syncytia that contain more than 3 nuclei. This one has 5 nuclei in the luminal epithelium. Further, as this diagram by Christiana shows, the histology of bovine placentation is just about epithelial choroid. It should be clean, it should be precise and easy to interpret. And that's not what we see during early implantation. It appears that the arrival of trophectoderm cells within the luminal epithelium really destabilizes the luminal epithelial barrier to the underlying stroma. Here we're looking at PEG immunostaining, and here immunostaining for SLC2A1, which stains the mononucleate luminal epithelial cells. And you can see a tremendous destabilization of that luminal epithelial barrier to the underlying stroma. It's no wonder PEGs show up in the circulation during this period of time. And when you look at individual days of gestation, you see some interesting things. Now, this is day 21, so we're pretty close to the beginning of implantation. And, it is assumed that we were just getting a few binucleate cells all along this interface forming and fusing with luminal epithelium to form trinucleate cells. But this is not a generality at day 20. Most of this uterine placental interface is still completely epithelial chorial. We just have these very focal, intense areas of trophoblast fusion, binucleate cell formation and fusion, like these are the sites that we saw in the previous slides. And when you look at by day 31, now this looks much more like I would have expected. We have these binucleate cells forming and invading into the luminal epithelium almost across the entire uterine placental interface, caruncular versus intercaruncular. But we see something else not predicted. There's luminal epithelium in some regions, although this is a pretty squamous looking epithelium. But there are other regions of the interface where you can't see any luminal epithelium. We've observed that with the sheep as well. There's no luminal epithelium here. And here we have a gland, which on both sides of the gland, we have regions where we've completely lost the luminal epithelium. It's taken over by this whole fusion event that's going on. We see this reliably at the openings of glands. And I don't know what it means, but I wonder if it's the beginning of the formation and sealing off of a relay. Then when we get to day 40 of gestation, we lose the luminal epithelial barrier completely. What we see in its place, for lack of a better way to describe it, because I frankly don't know what it is, it looks like a multi-layer thick heterogeneous population of PEG positive cells. They are not invading into the stroma and intercaruncular regions, but they're starting to invade into the stroma in the caruncular regions. And we think these are the beginning of cotyledonary villi. But note the cells leading the invasion into the caruncle, mononucleate cells. So are these the only mononucleate luminal epithelial cells left in these regions, or do they have another origin? And I ask that question because in interplacental regions, we see these SHMT2 positive cells dispersed within the trophectoderm, but they don't end up in the endometrial stroma because there's no invasion. But if you look in the caruncular regions, placentomal regions, you'll see that sometimes they're lined up right against the stroma. Sometimes they've invaded a little ways into the stroma. And when you look at that leading front of mononuclear cells, these cotyledonary villi, these are SHMT2 positive cells. So do mononucleate trophectoderm cells migrate to the uterus to compose the leading edge of these newly forming cotyledonary villi? When we get out to day 67, we see that the entire caruncular epithelium is now SHMT2 positive. So there's two possibilities. One is that luminal epithelium wasn't expressing SHMT2, but now it does. That's perfectly reasonable possibility. But there's also a possibility that the caruncular epithelium is actually composed of mononucleate SHMT2 positive trophectoderm cells that migrate from the trophectoderm layer to the endometrium to compose the leading front of developing and invading cotyledonary villi. I mean, this is complete supposition at this point. We have no strong evidence. But when you think about it, you'd think that a trophectoderm cell would be more migratory and invasive than a luminal epithelial cell. I'm going to finish here with some new immunofluorescence staining that was performed by Thina Manella, and this is a go figure picture to me because I did not predict that the binucleate trophoblast giant cells of cows would express MUC1, but here it is. When we took this picture, I thought this looked like the eyes of one of those little gray aliens, but Dr. Baser tells me this is a caterpillar, and I will defer to him. So I'd like to close by acknowledging our funding from the USDA, members from the College of Veterinary Medicine and Biomedical Sciences, members from the Department of Animal Sciences, and I leave you with this picture of the uterine placental interface of a cow that was published in Davenport et al. In 2023. Thank you.
All right, thank you so much, Greg. That was really wonderful. Just a reminder that if you have any questions, please feel free to tap in the Q&A box. But I'm gonna steal the first few questions if that's okay. So in the first part of your talk, this morphological change of elongating conceptus is quite striking, at least in the pigs. So what is evolutionary advantage for the conceptus going through such a dramatic morphological change?
I think it's their type of placentation, their epithelial corial and synepithelial corial. Pig is almost epithelial corial. Sheep is more syncytial. But that means that you have more cell layers to transport nutrients from the maternal vasculature to the placental vasculature. And so to overcome this, they do things like the intestine do. They increase surface area wherever they can. So they're elongating so that they maximize their surface area of attachment to the luminal epithelium because they're not going to invade inside of the uterus and have that easier access to nutrients.
All right, and also like follow up on that, it seems like you have discovered quite an elaborate network of metabolic circuitries, at least in the pre-implantation conceptus in the pigs. I was just wondering, like, have you looked at, across the elongation period, do those circuitries got tuned up and down, and also if after implantation, do some of them get shut down or like turned back on, something like that?
Yeah, we've looked at several of them later in gestation. One of the most interesting ones is glutaminase and glutamine synthetase. When you start to have the folded interface, some will be glutaminase will be in the tall columnar cells on the chorion, and glutamine synthetase will be down in the more squamous cells at the bottom of the uterine placental folds. Very, very distinctive pattern. You'll see some of these that will show up like your SLC transporters. Some of them will show up, particularly the high throughput ones, will show up in a relay and at the top, the uterine placental folds, which are areas of massive transport. And I think that is to maximize the transport across that. So yeah, these are effective all the way through pregnancy. And in fact, if I'd gone straight pig today, I would have put in some stuff that we worked on later, talking about the folds in a relay and all the changes in gene expression you see across that interface. But I wanted to please the cow and sheep people a little bit too. So.
All right, thank you. So now I'm turning to the Q&A chat boxes. A question from Rachel Garrity. Sorry if I mispronounce your name. Wonderful work, really interested in the immune aspect of the implantation process. Do you think that an increase in the interferon gamma from the conceptus could activate maternal immune cells like uterine natural killer cells that could potentially attack and destroy trophoblast cells of the conceptus?
Well, absolutely. I believe even in this type of placentation where we usually talk about it's hiding from the immune system, it's not. It's directing the immune system, and it's an inflammatory event, implantation. Even in a pig, you're changing that endometrial stroma. I wouldn't go so far as say you're activating natural killer cells that are going to attack the conceptus. That would be anti-productive. But definitely, I think I think that's why the interferon gamma knockouts don't make it through all the way through implantation, because you don't get enough stromal remodeling, you don't get enough angiogenesis and stuff, you don't get the things you need to happen without the interferon gamma, and that's all being mediated through those T cells, this semi-inflammatory but controlled inflammatory environment. So that's an excellent question. We absolutely do believe that manipulation of the immune system is key to implantation.
Wonderful. Question from Kathleen Karen. Beautiful presentation as always. Have you looked into the cellular mechanisms that mediate the uptake of extracellular vesicles in the luminal epithelial cells?
We have not. We have not. What you've got there is as much as we've got. So we haven't gone there yet. But
yeah, that'll be an interesting thing to look at. Yeah.
Right.
All right. Question from Abdul Wahid. Great presentation. What would be possible findings if we get samples from non-pregnant animal during this period?
For what animal?
Yeah, I think we probably need some clarification on this. Abdul, would you mind further clarify your question?
Plenty of evidence on the pig and the sheep that the difference is. We don't for the cow. This is one of the things that these cows are really expensive right now. It is hard to get them to give you the uterus out of their cattle, and we don't have the cyclic comparisons that we need yet. And we don't have the early pregnancy comparisons that we really need. So that's an open point that we're trying to fill in.
All right. Question from Rachel West. Thank you for giving such a fascinating talk. A paper came out earlier this summer documenting that endogenous retroviruses can facilitate fusion of the TE to the endometrial stroma in humans. Do you think endogenous retroviruses might be driving the formation of binucleate and trinucleate cells in large animals?
Oh yeah, and that's work from Tom Spencer and Fuller Baser. They've shown that clearly in the sheep that's occurring. Yeah, so that's definitely.
Yeah, all right. This is a question from an anonymous attendee. A very nice presentation. Thank you so much. Does interferon omega play a role in conceptus maternal communication in pigs or cows?
We don't know. We don't know. And the porcine conceptus produces an interferon delta, which is a type 1 interferon. But our group and Hak-Young Ka's group, we cannot clone that thing out. We cannot get it to where we can work with the darn thing. So we've been stuck with the interferon gamma thing, but gamma turns all these genes on. But there's literature out there that gamma will increase the effectiveness of a type 1 interferon and vice versa. So I don't doubt delta has something to do with it, but I know nothing about the omegas.
All right. This is actually a question from Hak-Young Ka. Wonderful presentation. This is a wonderful presentation, Greg. My question is there a possibility that the extracellular vesicles contain interferon D, it's in addition to interferon gamma, and I think D means probably delta, and affect the endometrial gene expression? And do you have any thoughts on how interferon gamma of extracellular vesicles interact with interferon gamma receptors on the surface of stromal immune cells?
Yeah, that's a great idea. We should be doing Western blots on those E3 preps to look for interferon delta. I never even thought of that. I don't know. I think these EVs can get these interferon gamma into the cell, and then it might just walk across that cell. And then how it is interacting with the basement membrane, if the receptors are or whatever, I don't know for sure. I don't know. But I think it's key for getting it into the cell. It doesn't necessarily have to be on bind to a receptor on the surface.
All right. Next question from Leonardo Geddes de Adred. Sorry for butchering your name. Apologies. In the bovine model, do you know if extracellular vesicles from embryos carry receptors or factors that modulate interferon, I assume this is tau, stability and bioavailability in the uterus, potentially prolonging its effect and serving as early biomarkers?
Well, I may be showing my ignorance. I'm not aware of a lot of data on the cow. I know there's data from the sheep that this is a possibility. And if you look back at that picture when I showed the interferon tau expression by the trophectoderm in cows, it's lighting up the luminal epithelium too. So it would be worth a look to see if there's any mRNA for interferon tau in that luminal epithelium, because that would be implied that the same thing's going on with the cow as what we saw with the sheep.
All right. There are also a few questions in the chat. Questions from Shakrog Patina. Again, sorry for mispronouncing your name. Does the horse Conceptus use the same method for increasing the epithelial surface, considering that it also has epithelial chorial presentation?
No, the horse does everything a lot differently, and they have a special invasive type tissues and stuff like that. So no, yeah, the horse is predominantly epithelial chorial, but it, as is always with the horse, it does everything different than everybody else. So no, it's not the same.
All right, cool. This is not a question, it's more of a comment from Saki Banu. Dr. Johnson is not only a great scientist, as we all know, he's also a great artist, Picasso of Texas A&M. So all the schematic images were created by him. Wow, great job, Greg. So another question from...
I'm getting fired by BioRender.
Another question from Shakrok. Do you think the T cells or other immune cells in the epithelial chorial presentation play a role in immune tolerance against the semi-allergenic fetus?
Almost certainly. Almost certainly.
Yeah.
I can't tell you all the specifics, but almost certainly. I'm not a believer that just because you're epithelial chorial, you're not using the immune system to help in the remodeling that is placentation. We already know that has to happen in humans. You just have no choice. But I think it happens maybe to a lesser extent in the livestock species, but almost certainly.
All right. A comment from Devendra Pathak. Excellent presentation with excellent quality images. Thank you very much. We all can agree. Another question from indeed Guzelok. Could porcine interferon gamma be recruiting natural killer cells and T helper cells and also reprogramming them to support pregnancy similar to its role in mouse implantation sites? I guess this question may be related to the previous questions.
Yeah, almost certainly. That's our hypothesis. It's recruiting T helper cells for sure. Haven't got any evidence on the natural killer cells, and we've had some trouble localizing natural killer cells there, although they're probably there. But most certainly that's our hypothesis, is the T helper cells are being recruited in. Yeah.
All right. Another question from Cecilia Constantino, Russia. Excellent presentation. So do you think if pig is knockout interferon gamma receptor, pregnancy would still be established because of the extracellular vesicle transport?
That's a really cool question. I don't know, if you don't have the receptor, you probably won't survive up even to that point, But yeah, it could be. It could be.
All right, cool. Thank you so much for all the questions from our attendees. And I think this is a perfect time to conclude our webinar. Thank you again, Greg, for this wonderful presentation. And thank you everybody for participating. And we'll see you in the next webinar. Thank you. Bye.
Well, thanks for the opportunity to speak. And hi to everybody. I'm just going to go now.